focus will be on carbon dating because it is, yes, a widely known field. But then, if I just show you a few pictures, like this is a burial, there are bones, some of them fossilized, but mostly bones, pieces of charcoal, a leather slipper, and these are in different museum collections, a fragment of textile, and paper. So, the one thing that is common in all these objects is that they have carbon content in them. All of them have organic material present in them. And if you have organic material present in any object, if there is carbon, you can essentially date it. At least this is what I used to think when I read carbon dating for the first time in class 7th or 8th, that if there is carbon, you can date it. But the objective of this lecture will be to understand that carbon dating has its own limitations. It cannot work in every case. It has its own drawbacks, demerits, but conclusively, we have to understand why carbon dating is still important in archaeology. So I'll just down the basic content of this lecture. We'll start with discussing what isotopes are, why they are important in archaeology, how they are used in archaeology, and what are the principles of carbon dating, what are the problems faced in carbon dating, and how archaeologists use different methodologies, use different ways to go around this problem. And then together we will arrive to a conclusion and evaluate, is carbon dating still useful for archaeologists? And this is what primarily the lecture is going to be about. We chose to focus only on carbon dating because there is a lot to be covered when talking about isotopes or bioarchaeology. And to make the lecture more coherent and easier to understand, a better introductory lecture, we are therefore sticking just to carbon. Um, so let's start with isotopes. This is nothing but just basic 10th class chemistry. So any matter is composed of individual units called atoms, and atoms have three parts: protons, neutrons, and electrons. The protons and the neutrons form the nucleus, the center of an atom, while the electrons revolve around the nucleus. Atomic mass refers to the number of neutrons and protons present in an atom. That is what the atomic mass is, while the atomic number constitutes of the number of protons present in any atom. This is important because an isotope is essentially a kind of an atom of any matter which has similar atomic number. That means the protons present in the different atoms are going to be same, but the number of neutrons will differ, and because of which there'll be difference in the atomic mass. This is what an isotope is. Very basic, but important to understand when starting with radiocarbon dating or isotopic studies. So let's discuss about carbon and understand the three isotopes of carbon which are important in archaeology: C12, C13, and C14. So as you can see, the atomic number of the three isotopes is same because of the same number of protons and electrons. The difference occurs because of the number of neutrons, 6, 7, and 8, and therefore the corresponding atomic mass is 12, 13, and 14. Now, these three isotopes are the ones that are used in archaeology, although there are a number of isotopes of carbon. 14 of them are known. And in fact, any sort of matter, any sort of element has its own isotopes. Because of the difference in the atomic mass, there are differences in their properties as well. So C12 and C13 are stable isotopes. By stability, it means that they do not decay over time. C14, on the other hand, is an unstable isotope, which means that it will decay over time at a constant rate. And this is precisely the mechanism behind radiocarbon dating, or C14 dating as we call it. It is the dropping of the level of C14 that is seen in ratio with the level of C12 and C13 to understand the age or date the age of an organism or object. So we'll go into the details of radiocarbon dating, but before that, I would just take a few minutes to talk about the other isotopes used in archaeology because they are important. So, nitrogen is also used in archaeology to understand what a person's or an individual's diet could have been. So, plant or animal diet or marine diet because it addresses the increase in trophic level. So, plants are eaten by animals, animals are eaten by humans, and humans also eat plants. And so, this elevated composition of nitrogen can tell us about what a person's diet could have been, person's or animal's diet could have been. Oxygen isotopes are used for understanding climate and temperature in the contemporary period and could also therefore help us in understanding where the organism could have resided or where it could have come from. And since we're talking about origin, it's important to mention strontium, which is used for individual biography because it can actually tell uh, through analysis of different bones in human body from where a person could have come, where it could have lived in the later phases of its life. So, yes, bioarchaeology offers a lot of potential, and this is something that, like a diagram that I could not resist but putting it here because let's say you're an archaeologist and you stumble on a bone randomly lying at a site. There are a number of things you can predict from that bone theoretically, and if it has organic material present in it, you can tell about DNA, you can tell about carbon, oxygen, and also through inorganic material, you can tell about provenance, diet, body temperature, etc. So now we'll be just focusing on radiocarbon dating. So how does radiocarbon dating work? It's a very simple explanation. So when cosmic radiation mixes with nitrogen isotopes in the atmosphere, C14 is created. The C14 isotope is created. This C14 isotope is present in the atmosphere along with the C12 and C13 isotopes, and together all of them form part of CO2 or carbon dioxide, which is, as we know, fairly simple process of photosynthesis absorbed by plants, and plants intake C14 isotopes along with them. These plants are eaten then by animals, by humans, humans eat animals, and through this chain, the isotope ends from one organism to the body of other organism to third organism, and an entire cycle is created of movement of C14 from atmosphere to the lithosphere and to the hydrosphere as well, because we are not forgetting the marine animals. What happens next is when the organism dies, so when the tree dies or when the animal or human being dies, its body starts, the C14 present in its body starts depleting. Why C14 and not C12, C13? C14 is radioactive. So the level of C12 and C13 remain constant, and so you can see the C12, if this is, let's say, a sweet 12 and this is C14, it will remain constant as the years pass by, but C14 will keep depleting. And it is the ratio of this difference that tells us the age when an organism would have died, the years that would have elapsed since when the organism would have died. So essentially, if an organism would have died, let's say, 10,000 years ago. So the number of C14 still present in that body will be calculated, and through that, the age of the organism would be predicted. So here is something also important that has to be understood about C14, that the half-life of C14 is 5,730 years, which is the age since then the C14 isotope would reach its half decay age. And this is like one of the properties and also one of the disadvantages of C14 dating, that you cannot go back more than 50,000 years ago. So in some cases, it has been speculated that you can go back up to 60,000 years ago. But beyond that, C14 cannot work for dating. So there's a limitation right here. Um, and therefore, in archaeology, carbon 14 is important, but when we talk about subjects like paleontology, or as we popularly call them, the study of dinosaurs, where the age is definitely way longer than that of humans, um, other forms of dating, other isotopes like uranium, thorium, can be utilized. So, yes, there are other isotopes for dating as well, some of which are potassium, argon, uranium, thorium, and others, which have a fairly larger span of dating the individual, the organism's life. But for C14, it's 50 to 60,000 years ago. I would like to just mention a few principles that govern radiocarbon dating. They are important because when we talk about these principles, that is how we come to know about the problems in the dating. And these were published in a paper in 1984 written by Therese, which is kind of very old, but then these principles are fairly important. So the first thing is that in order for radiocarbon dating to work, the production of radiocarbon has to be constant for the last 50,000 to 100,000 years. So and it's not just the production, it's also the absorption or uptake by organisms, the exchange that takes place through different spheres. It has to be uniform, and so has to be the decay rate of radiocarbon. The other principle is that no sort of young or old carbon should be added um, in your sample. This is also important because this points to a very, very important problem that takes place in radiocarbon dating, and that is contamination. And finally, fractionation should not have occurred. So, fractionation, I'll be talking about fractionation in detail in a few minutes. But what fractionation is when any isotope gets separated in different compounds, or if the compound remains the same, or the substance remains the same, but the phases are different, that process is called fractionation. And this problem will also be discussed with a case study. So just hold on to it for a bit. Yeah, okay. So let's talk about the first problem. Is the production of radiocarbon constant in the atmosphere? That radiocarbon is formed in the atmosphere uh, with cosmic radiation on one hand and nitrogen isotopes on the other. So if there are problems in either of this, the level of either of this goes unbalanced, there are going to be problems in carbon 14 production. So interstellar activities like supernova bursts or movement of stars or any other thing can cause problems in the production of C14. Even activities of sun, so like occurrence of sunspots, which are areas of high magnetic field and they appear darker, as you can see in the picture, like these patches, can also affect C14 production. Short-term oscillations, change in number of neutrons, and variations in solar energy can also lead to problems in C14 production. And the other thing is to understand how much of solar radiation or cosmic radiation reaches Earth, which is determined by the magnetic field of Earth. And this might shield cosmic radiation. This was studied by scholars long back in time, and it was suggested that this change in Earth's magnetic field can create a lag of almost 5 weeks, which if we think about practically is not really a concern. But then something that has to be studied in terms of the production of radiocarbon is that these are fairly older papers. And I have to say this here that a lot of research has not been done on these aspects of radiocarbon production in recent years, which is an important area of research because we need to understand since galactic activities, solar activities are also changing. And another important thing to consider here is that we are talking about dating organisms for a period that go back to 50,000 years, which means that such changes would also have occurred in this huge time frame. So, what uh, cosmic flux could have taken place in these 50,000 years also need to be considered or thought about when dealing with this problem. But however, scholars have also suggested that the impact of these activities on C14 can not be overstated, which again is debated. So I'll leave this debate right here and venture into other problems. So we have C14 produced in atmosphere. Now C14 is going through different spheres. So the exchange of C14, if we remember the principles of radiocarbon dating, it has to be uniform or constant. But does it happen? So Swiss effect was something that was identified in 1955, literally 5 years after radiocarbon dating was suggested by Libby, uh, that the burning of fossil fuels creates larger amounts of CO2 in the atmosphere, and this reduces the level of C14. This happens because combustion of fossil fuels produces carbon dioxide, and carbon dioxide does not have isotopic content in it, and therefore the C14 level would fall, and this would create an entire cycle of imbalance between atmosphere, lithosphere, and hydrosphere. To understand the gravity of the situation, I would like to cite this paper that was published in 2015 by Gravin, where he said that if the fossil fuel combustion continues happening at the same rate that it has been happening, by 2050, the C14 to C12 ratio of the fresh samples would be almost that of 1050 CE. So you can see that the error is of 1,000 years. And any object, like if an object shows an error of dating of 1,000 years, it can lead to huge archaeological problems in dating, let alone dating being imprecise. It is going to be totally wrong. Another thing that has to be considered here is the marine reservoir effect. Basically, what marine reservoir effect is that the carbon that is consumed by organisms in water is often older than carbon consumed by organisms in plants. And this is also to do with the trophic level. This is also to be done with the absorption of carbon dioxide by terrestrial plants and aquatic plants, and then these aquatic plants are eaten by fishes. So simply it means like aquatic plants, aquatic grasses, they absorb carbon dioxide, they are eaten by fishes, fishes are eaten by larger fishes, and then they are eaten by human beings. And so when you would date these human beings, the dates would come out to be older than the period of existence of that human being. And this is what the marine reservoir effect is. Other than the Swiss effect, it has also been suggested by certain scholars that the Swiss effect has been overcompensated because of nuclear activities. Nuclear activities will generate isotopic content in the atmosphere, which will also disturb this constant state that was suggested for radiocarbon dating. And non-anthropogenic activities like meteoroid activities, volcanic eruptions can also cause disruptions. Minute change of 1 to 2% like can occur because of eruptions, but if there is a sustained effect that is taking place, so like combustion of fossil fuels, that can cause a change of 10%, and this 10% change can give an enormous difference in dating again. So let us leave alone these problems and now focus on something which is very, very important, because this is something that happens not just in radiocarbon dating, but any sort of isotopic analysis, DNA, and largely in bioarchaeology, and that is contamination. So, contamination can happen in a number of ways. It can be true contamination or admixture. True contamination is caused by natural factors, while admixture can be because of human negligence or human error. But the good thing here is contamination can be removed and it can be managed. At least on paper, it can be done. But does it happen in real life? Let me explain this through some pictures. So this is like a stratigraphic context that you have to excavate. Let us say this is the context that you are excavating. And you can see that there are different soil layers, which means they're obviously of different dates. And say, let's say I discovered bone in this part, and the bone already would have some sort of contamination that would occur because of acids present in the humus, organic material that is already existing, other artifacts that are present in the layer, and twigs, branches, or dead roots. All of these are different sorts of mixtures that will be present in the layer, and this will affect the dating of my sample. And so, as an excavator, I have to be very careful about noting down the layer from which my object is coming from, something that we all know in archaeology. We have to be very careful of understanding what are the other objects also present in the same layer and how they could impact my sample, how they could impact its carbon content. Like we are talking about carbon. Carbon is fairly abundant, not really abundant, but it is present in almost all circumstances. And we have to be very careful that when we are sending these samples, all sort of dirt, these twigs and branches and roots and everything has been removed clearly. And let's say like you come across this. So this is a burial that you might encounter. And so you have to be very careful about cleaning it. And the true contamination factor is this uh, where you would be, it is done mostly in laboratories, but admixture is something that occurs in case of excavations. So you clear all the manual dirt and everything that you can, and then you send the sample for lab. When you send it for lab, um, let's say because it has been thousands of years since you excavated the object, and so there'll be deposition of silica, carbonate that you can dissolve through acids like sodium hydroxide or HCl, and these are what are used in laboratories, dilute solutions of the same acids, and in lab you would be treating this true contamination, and then you would be using your sample and you might put it in the state of art machine that you have. So it can be in carbon dating, we use accelerated mass spectroscopy. There are even better or even more precise equipments like TIMS or ICMPS, and you can use them for analysis to get more precise results. But the at the end, the thing is that is the problem of contamination solved? So there are no standardized procedures, even in labs. So which acid should be used? Which acid should be used in which circumstance? Uh, how much of dirt has to be removed? How much of dirt has to be kept to keep the context intact? There is no standardization that really exists in different laboratories among different researchers. And let's say that in your sample, contamination has already occurred. You publish the results, and there are, let's say, two pieces of charcoal, and because of contamination, even if they are found in the same context, we get a difference of dating, let's say, of 2000 BCE and 1000 BCE. And this is a hypothetical example. And so you would calculate the mean age and say that the layer could be dated to around 1500 BCE. And you publish the results, but you did not talk about the contamination that is there. And so will you be re-verifying the results that you have published? Again, all these spectrometers are expensive. Can they afford it? Can they be afforded by all laboratories? And if not, then are we actually going to rate more precise samples, or are we going to rate the more imprecise samples? And finally, the problem of preservation. Now, this is like one of the most challenging parts of radiocarbon dating. Earlier, samples were taken in grams and then they were burnt to accelerate the ions, and ions were then put into a fuel kind of a thing, and through that, the movement was registered in a magnetic field and the dates were calculated. So carbon dating is a destructive mechanism. That means the sample that is used for dating will get destroyed eventually. Yes, researchers have been able to reduce this sample from grams to milligrams, but then again, will your sample be preserved? The last one is accountability, which is also related to the second point, is that if the results published have contaminated samples, if there is a problem in dating, then who is going to ask questions? Who is going to revisit these studies? Will the researchers themselves come up with revision of their own published papers? So the problem of contamination can not really be solved. Yes, on paper, as I said, it can be solved, but then there are other things to be considered. Johnston in 1973 explained the problem of radiocarbon dating and what contamination can do to it. And I'll show this graph to that was there in his paper to say that I'm not over exaggerating the situation. If there is a contamination of 1% in your sample, so if the true age of your artifact was, let's say, 60,000 years ago, it will come down to 40,000 years ago. So the sample will show age 40,000 while the real age is 60,000, which is a difference of 20,000 years. And if the contamination goes above and it is, let's say, 10%, then the age shown would be reduced to 20,000, which is a difference of 40,000 years. 40,000 years. Think about this situation this way. So Neanderthals were present in Europe for almost 80,000 years. And if there is 20% contamination in your bone samples, so maybe the Neanderthal skull that you would be studying, if it has that 20% contamination, you might think that you are looking at the most recent or the last of the Neanderthals present, but it might be the earliest of the Neanderthal present. So yeah, that's how much contamination, even the minutest of contamination, can affect radiocarbon dating. This extreme fluctuation is called something like outlier effect. And with this, we are done with the problem of contamination. But other than this, there are other problems in radiocarbon dating as well. And by other problems, I do not mean that these are secondary problems or are not as important as the other problems. Calibration curve, as you might have heard about it, is another one of major issues in radiocarbon dating, because the dates obtained have to be converted in calendar years, which is not easy for researchers to do that. There are calibrations that have to be done, there are adjustments that have to be made, and in a number of cases, they are not pegged against any set reference system. Even if they are, they might not be published or clearly discussed in the studies that the researchers will be showing to the world. And thus, communication, as we come to it, is another problem, is a big issue. And if there is a lack of transparency that exists amongst academicians themselves, so can we really say what the public would know about it? So, with this, we come towards the end of the problems in radiocarbon dating. And since we have criticized radiocarbon dating enough, now is the time to deal with a few case studies to understand how archaeologists deal with the issue that are present in dating. So the first paper that I wanted to talk about is this study that was undertaken in 2007. And it was done in northern Iceland of a period of around 900 to 1000 CE. And here the problem was that my with by with Nisbet, there were bones of horses and humans that were found, and both of them were dated. It was found that the dates of the humans from the stratigraphic content was older than that of horses. What does it say? It is a marine reservoir effect, or in this case, it was fresh reservoir effect as well, because whatever marine organisms were consumed, they would have been from the largest sea and also from freshwater contexts. So, to understand this imbalance of C14 that is there in the atmosphere and the hydrosphere at that time, 900 in 900 CE, it was important to make a list of the marine reservoir effect. That means to quantify this difference that exists for organisms of the same period but consuming different diet. And therefore, what the researchers did in this case was they took cattle bones as emblematic of the terrestrial or atmospheric C14 and mollusks for representing the marine C14, and dating both of them together, they were able to come at like a list of differences that exists in the C14 levels, and they were able to create an MRE list, because of which they were able to successfully date the period and the samples. So, okay, I'll just go a bit more into this case study. Um, there was a problem that exists in the same stratigraphic context, and radiocarbon dating showed different results, which was because of this marine reservoir effect. What was marine reservoir effect? Marine reservoir effect happens when organisms from the sea show a later age, and these organisms, when consumed by organisms on the earth, show an older date than of their contemporary organisms. In this case, it was humans. And so terrestrial C14 levels and marine C14 levels, which have a difference, which basically MRE is, was calculated, it was presented, and through this, the period was successfully dated. So, yeah, another important thing in this study was that researchers were able to also understand diet of pigs. Now, pigs, because of the increased nitrogen isotopes and also because of MRE, can be shown to have an omnivorous diet, and this was suggested through the study. The next paper talks about fractionation. So, fractionation, as I said, is a problem. While we left earlier, we'll be discussing it here now. So, it has been found that in case of plants, plants are usually not considered as the best samples for radiocarbon dating. And the reason for this is that in the same tree, different parts can have different dates. Why does this happen? This happens because certain plant species like oaks or junipers have trees that go back to hundreds of years. That means the trunk, the central part of the trunk, would be fairly older than the outermost layer of the trunk, fairly older than the young branches or the twigs that are present, and therefore there can be a difference of dating of the same sample of trees in different periodical contexts. This is what fractionation is as a problem encountered in radiocarbon dating. And what I just explained is the old wood problem that did occur in this case as well. So, lack of plant association with the archaeological context, different rates of fossilization of plant parts, fractionation, old wood problems. These are the usual research problems that are faced in dating of archaeological plants. So, in order to deal with this problem, in this study that was published in 2017, the researchers said that let's emphasize first of all on classifying these trees. So, which variety of tree is present in which region? And they did it for Neolithic sites in Poland, Slovakia, and Hungary. Through C14. And what they were able to do, that they were able to identify plants which were older or which would not have been present in the Neolithic times. Also consider that this is Neolithic period. This is the time when certain plants species will be domesticated. Example, hurled wheat. And so you can understand which plants would have been actually grown in that region, which plant would have come from a different region. And when you are sure about which plant you're dealing with, which part of the plant you're dealing with, you can attain dates. You can actually use dates that make sense. So they used charred seeds, twigs, and branches for considering for dating the period, because they actually represent a season or the time when they are talking about it, when they are talking about the tree, but not the main cortex, not the trunk or the central part of trunk for dating, because that would have given an older date. And so the obtained dates were then also juxtaposed with radiocarbon dates of bones and archaeological samples that were obtained from these Neolithic sites to ascertain a wider line of evidence. So, in this case study, through this case study, the researchers emphasized that we have to also think about which plant one is talking about or which plant one is dealing with when we are talking about dating these plants. And finally, the last case study. This case study is something that does not really deal with the problems that I had mentioned earlier, but it tells us about the scope of radiocarbon dating, which is fair enough since we have been criticizing radiocarbon dating in the last 30 minutes. So, what the researchers did in this 2021 study was that they took samples and they dated these samples two different kinds of samples, and through that they were able to situate the sample in their historical context. What were the samples? So the samples in this case were paintings that go back to the 16th century, 17th century, and one of them which goes to the 19th or the 20th century. So all these paintings were housed in private collections, and the problem of private collections is that we do not know who bought these collections, who the artist was, where was this painting made, where was it commissioned, how it ended up in the collector's office. Maybe they know, maybe they do not know, maybe they do not tell about it. And so, since the paintings did not have any authorship or any name of the painter, they decided to date them. And for this, they utilized two different approaches. So they used carbon dating for dating the paint layer and then for also dating the binder. And then taking both the dates in consideration, they were able to situate the painting in its historical context. And through this, while they were able to situate the painting in the historical context, it's also important to say here that they also mentioned a demerit of their own study. So, in case of the 19th or the 20th century painting, um, the binders that are available, they are not made of organic material, they are made of synthetic material, which made it difficult for them to situate this painting in its historical context. And this becomes very important because in this case study, researchers did not just go over the top ahead that, oh, we are going to date art pieces with radiocarbon dating. They took a step back and also mentioned the problem or the limitation they encountered in their study, which I feel is very important to admit. So, finally, we arrive at the conclusions. So, the strength of radiocarbon lies in the fact that it does have a wider application. Organic material can be dated through carbon, are dated through carbon, and it is since a fairly wide practice. It has a wider application. It makes it easier for researchers to compare objects of different contexts and to look at the relationships between them in terms of dates. The cost has also been reduced, and that is because of better technology available. Although, yes, the spectrometers are still expensive, yet the cost can be micromized through a better kind of TIMS or NCICPMS uh, spectrometers. Comparability is there, acceptability of radiocarbon dates still exists, but there are weaknesses as well. So the first weakness is that, as mentioned earlier, that the time span of radiocarbon dating is just 50,000 years, which is fairly less. But the other problem that we have discussed in detail is contamination, which still exists. And as also pointed a bit in the last case study, there is less transparency in talking about what are the samples of research that were taken in by researchers, how these samples were calibrated, or how these research was published. And so many of them that may have had contamination have never been rethought or reworked upon. So, as archaeologists, what can we do? Definitely, we have to go for a more interdisciplinary approach. So, like we saw in the fractionation problem, do not just focus on radiocarbon dating. Also, take into consideration other archaeological artifacts that you see across in your studies. Use multiple isotopes. So, in a number of carbon dating methodologies, nitrogen that was used in the Iceland problem to understand diet was added to give more evidences for the diet and the MRE effect. Therefore, to go interdisciplinary, to focus also on other traits like morphology. So, understanding what the tree species could have been, understanding what the stone tool looks like, understanding its patterns are important. And if we are dealing with the historical period, then definitely literary objects have to be taken in consideration. So, we have to adopt an interdisciplinary approach and not rely solely on carbon dating as if it was a sort of miracle that is going to provide us all the solutions at once. The other thing is for the researchers to be aware of their own biases and limitations and share them with their results. Um, Contaminations definitely, and here I would also like to like quote um, this sentence in a 2005 paper that radiocarbon laboratory dates the sample we submit, not the feature we want to date with that sample. So be very clear about the sample that you are taking in. So the sample, remove any sort of physical contaminations, any sort of chemical contaminations. Be practical, that can it yield the dates that we have in mind? Can they not? Can it be used in a specific context? Can it not be used in another context? So let's talk about paper. The trees that are used for making paper, when were they cut? When were they cut? And that would be from where the radiocarbon dating starts. Not, it will not date the paper exactly when that object was written on or when that object was used, but it will date the time when the tree was cut. So being aware of our own limitations is essential in any kind of radiocarbon study. That's it from my end. And this is selected bibliography. And thank you everyone for joining me for this brief lecture.